What I'll talk about today will be really methods, techniques, and ways that we can think about the effects of light on humans. And I will kind of make it, in a way so that there will be technical information, but it will also still be accessible to those who do not, for example, have a background in, say, radiometry, photometry, or lighting science. I just want to quickly highlight how my team is set up. We currently have 3 sites that are working together in one research unit on various areas. If you are interested in hearing more about our work, then tscmlab.org is our website. This is part of the team. We spent some time doing a lab retreat in 2024 on Zugspitze, where in fact, some of us had, because of altitude sickness, some issues sleeping, probably something that we want to investigate further and see how we can counteract it. And more broadly speaking, the areas of research that my team is working on include mechanisms underlying non-visual photoreception, circadian physiology in health and disease, real-world light exposure and its effects, behavioral technological interventions, and ecosystem development. And what I'll talk about today will be mostly on our knowledge and our insights on real-world light exposure and its effects, but then also ecosystem and development. So we'll start with a little bit of an introduction of the fundamental underlying biology of what we know about how light is processed in the human eye and in the human retina. So I think it is probably not an understatement or an overstatement to say that over the last 20 years or 20 plus years now, our understanding of the human retina has changed significantly. We've known for a long time that there are different cells in the retina that do different functions. And the cells that have been studied in the domain of vision science and sensory biology and visual neuroscience, most predominantly are the cones and the rods. The cones and the rods are what we consider the canonical or the classical photoreceptors that help us really see. And by seeing, we usually mean vision and visual perception. The cones come in 3 different flavors or 3 different types that prefer different wavelengths. And they are highly optimized to capture light under daylight or daylight-like conditions. The rods are different photoreceptors. They are, for example, not present in the center of the retina, which is where under daylight vision we have the highest visual acuity. There's only 1 class of rods, and they are a lot more tuned to dim light conditions. And so the rods and the cones really cover an enormous visual range or an enormous dynamic range of light intensities that are present under natural conditions. And it's kind of during twilight, so during dusk and dawn, when both rods and cones are active, when kind of there is this transition between what we call the photopic regime and the scotopic regime, where photopic means daylight or daylight-like, and scotopic is everything that's mediated by the rods. So I've said a lot about the rods and the cones, but in fact, I also mentioned that our understanding of the human retina has changed significantly. And so this is where we introduce the so-called intrinsically photosensitive retinal ganglion cells. This is abbreviated as iPRGCs. They are types of neurons in the retina that are directly light sensitive. And so basically, when we're looking at this kind of schematic diagram of the retina here, you have the layer of photoreceptors and then layers of neurons. And the sort of last layer of neurons before this information leaves the eye through the optic nerve is the retinal ganglion cell layer. And so it turns out that a fraction of these retinal ganglion cells are light sensitive independent of the rod and cone input. We know from various studies that the rods and cones do provide input into the iPRGCs, but they also have an intrinsic capacity to signal. So when you isolate them through experimental techniques, they will still signal and encode ambient light intensity. And of course, and this is, we'll come back to a little bit of measurement geometry later, all light that is relevant to the effects that we'll be talking about here go through the lens and then are imaged on the back of the eye on the retina. And so this is a cross-sectional diagram. So melanopsin was discovered in the skin cells of the African clawed toad, which basically changes its coloration or changes its shading depending on the environment. So this is an animal that changes what it looks like, how light it looks, depending on the surrounding environment. And that is mediated by this photopigment, this protein melanopsin. And this was really the discovery of melanopsin that was kicked off, and it turns out it is also present in the mammalian retina. Many studies have demonstrated that it's actually quite well preserved in terms of the spectral sensitivity. And of course, we humans also have melanopsin. And this is really where I think we have an interesting sort of convergence of research findings. So the discovery of melanopsin and the discovery of the iPRGCs was almost at the same time as a lot of the basic findings of how light impacts human physiology and behavior in a non-visual kind of sense. So what we know about the photoreceptors ultimately also determines how we should think about measuring light for understanding the impact of these photoreceptors. And so in this diagram, I'm showing you a plot that we'll see in similar ways later on as well. On the x-axis, we have wavelength, where lower numbers correspond to short wavelength and long wavelength correspond to larger numbers. And on the y-axis, we have the relative sensitivity of these sensory photoreceptors, of the cones, the rods, and the iPRGCs. LMS are the cones, melanopsin is the melanopsin component of the iPRGCs, and R here corresponds to the rods. And what you can see here is when we're looking at the spectral sensitivity, which is really the preference of these different photoreceptors to different wavelengths, we see that all photoreceptors cover basically an enormous range of wavelengths. And this is quite interesting because the photoreceptors are severely overlapping, right? So you see, for example, that the melanopsin and the rods are quite overlapping, as are the M cones, as are the M cones and the L cones. Now, I think this is quite interesting because, the fundamental biological discovery took place in the late 90s, and then there was a lot of neurobiological work that tried to tease apart, what's the role of melanopsin and so on. And really, it's quite remarkable that within 20 years of the discovery of melanopsin, we have converted this knowledge of the melanopsin containing iPRGCs into an international standard, which is the CIE system for metrology and optical radiation for iPRGC-influenced responses to light. And so this is going to play a role later on when we're looking at quantification, but I just want to make sure that it is clear that basically where we are in terms of measuring light for the non-visual effects of light is directly related to how we understand the physiology of what's going on in the retina. And so I usually call this physiologically relevant measurements because you're measuring light, of course, with respect to a physiological mechanism that you're trying to understand. I mentioned that there's iPRGCs that express melanopsin. What do they do in sort of the entire human being, right? What is their function? And what we've known, or what we've gathered evidence for over the last decade is really that they drive the primary drivers of what we call the non-visual effects of light. And I'll give you a couple of the examples here. The first very well studied phenomenon is melatonin suppression through light. So light suppresses the production of evening and nighttime melatonin, and it follows a dose-response curve where basically the brighter the light is as seen by the iPRGCs, as seen by melanopsin, the stronger the suppression. At some point there's saturation, right? So if you're bright enough, then you'll see the saturation. Different studies have also examined the circadian effect of light. So these are studies in which researchers investigated to what extent light in the evening or at night can shift the circadian rhythm. There again, we have a similar dose-response curve where I think it's pretty straightforward. Increases in light intensity lead to a stronger parametric response. And then we have, broadly speaking, classes of neurocognitive effects, which here is measured as Karolinska sleepiness scale scores, where, again, we have alerting effects of light, of evening light, that are driven, again, by melanopsin. And so there's evidence that, of course, as you increase light intensity, it will stimulate melanopsin. It won't do so in isolation, but several analyses have shown that really it's the melanopsin component that primarily drives these effects. So when you put all this together, we have a very nice joint dose response curves that was summarized by Brown et al. in an expert group that I was privileged to be part of, along with Christian Kajochen and Mirjam Münch and maybe several other people in the audience, where we put together the evidence and try to understand what can we do with these studies in terms of recommending specific light levels for the daytime, the pre-sleep, and the sleep environment. And so this is really kind of has changed a lot, I think, about how we think about the impact of light. It has generated, I think, an interest in understanding how can we optimize lighting conditions, and is really currently a good basis to make recommendations on. Of course, for any studies that are done in the laboratory, there's always the question of how do they translate into the real world, which is, of course, something that is often or is an important area to consider. And when we're looking at the general research landscape, we have a lot of laboratory studies. And then when we're looking at real-world evidence, there's not so much evidence yet. But of course, the group in Basel, my group, and other groups are working to generating this evidence. And again, this is a key reference that I think is really the framework for how certainly my group thinks about light. It's the recommendations paper that was published in PLOS Biology. However, if we want to understand the non-visual effects of light exposure, we need to understand light exposure first, right? So we can't skip the step of looking at only the output. We need to look at the input. And this is what I want to talk about today, is how do we do these measurements? What are some practical techniques? And then also, this question of real-world relevance, how do we connect it to the real world? So, and I basically have prepared something here that gives kind of a three-step process for documenting, for measuring, for characterizing the effects of light. But I think this is a very useful thing. You always want to look in the literature to see, has someone done similar work before? And SLTBR, which is now called SLRCH, this is now the Society for Light Rhythms and Circadian Health. Then the Society for Light Treatment Biological Rhythms had a bulletin for a while, and which is available online. And so there's a paper or an article in the August 1991 bulletin that is Standards for Specifying Parameters of Bright Light Stimulation for Therapeutic Applications, a proposal. This is, of course, in the context of light therapy, but I think we can still learn a lot about how we should think about measuring and quantifying light. And this is quite interesting. This is from the text. In contrast to detailed analysis of clinical response presented in the literature, however, there has been minimal attention to adequate specification of stimulus parameters and exposure conditions, i.e. standards for the lighting systems used. And this is, of course, where we can learn from the work that was done 35 years ago and kind of see how can we build on that. The same article contained basically some minimal information that was relevant for documentation, including exposure geometry, illuminance, and so on. But unfortunately, this proposal never really caught on. This is not very well cited. But of course, it's useful to know that whenever you have an idea, someone probably had a similar idea. And it's nice to see also that these ideas have been around for a while. I want to propose a framework that helps us answer this question of how we should measure and quantify the effect of light that has really three steps. The first one is measuring, the second one is characterizing, and the third one is reporting. And so really, as you are either working on studies that use light as an intervention, or if you're working on evaluating lighting systems, or in some way are interested in understanding the potential impact of light on human physiology, then these three steps will be very common to that enterprise. So we'll start with measuring, and I think it's of course, accepted in our field that what we care about is light stimuli as characterized from the perspective of the observer. What we want to know is the dose that a person is receiving, and not a dose that, for example, a tabletop is receiving. And of course, there might be geometric relationships between these two, but if we're thinking about exposure, we need to understand exposure from a human-centric kind of way. There's a second component here, which is in this measurement statement, which is the spectrally resolved way, right? What we want to make sure is that we can capture the light, not in terms of illuminance or color temperature on its own, but in terms of spectrum, right? So the measurement should be measured from the perspective of an observer in a spectrally resolved way. We then want to convert the spectral measurements into physiologically relevant metrics and quantities, amongst which the CIE SO26 is the international standard. And by physiologically relevant, what we mean is really something that is directly related to the mechanisms that underlie ultimately the response that we're interested in. And then finally, and I think this is a key component, not so much for understanding the effects of light and quantifying them, but it is for making sure that we report them so that when we publish papers or when we document it, it is easily accessible in a standard and harmonized way. So let's take a look at measuring spectra. We know from physics and physiological optics that the spectrum is for, of course, removing temporal and spatial factors, is the most complete physical representation of light. So again, I've already shown you this diagram. On the x-axis, you have wavelength. On the y-axis, you have power, in this case, instead of sensitivity. Shorter wavelengths kind of are bluish, longer wavelengths are reddish. And what we can now plot is the spectrum of light. And so what I'm plotting here is a characteristic representation of a daylight. This is daylight at a correlated color temperature of 10,000 Kelvin. This is what the spectrum looks like. It is downsampled to, I think, 5 nanometer spacing. But what you can nicely see, actually, even from these plots, is there's these little dips. And if you have high resolution measurements of daylight, in fact, you see what's called the Fraunhofer lines. These are the little dips in the spectrum that correspond to the absorption of specific of light, of optical radiation through specific molecules in the atmosphere. So here, this is, of course, the sun is a radiator. And when the light that comes from the sun is filtered through the atmosphere, you get these characteristic daylight spectra. And what is added here is a curve that corresponds to a black body radiator of the same correlated color temperature. So if we didn't have filtering in the atmosphere, then this is what such a spectrum would look like. This is in normalized units. Of course, we cannot convert this into lux or anything like this, but it's just a way to show the spectrum. It's important to keep in mind that when you can, which you should, measure the spectrum of light. And what's important is, of course, that spectra come in different flavors in the world, right? So here we have 3 different daylight spectra that have different correlated color temperatures, one at 4,000 K, one at 6,500 K, and one at 10,000 K. And these, of course, change in how orange or bluish they look. And it turns out, in fact, that when you're looking at, for example, twilight, you do have sort of the short wavelength boost in intensity, which leads to very high correlated color temperatures at or before or after dusk and dawn. And so these are the daylights. You can measure these now, but of course we also have artificial light, and so different types of spectra look different. These are some standard illuminants, standard illuminant A, F12, and a white LED. They have a similar correlated color temperature, but what you can appreciate is, in fact, that they have very different spectra, right? So they have the same lumen, the same correlated color temperature, but are spectrally very different. Now, there's a fundamental thing that I think is really important to recognize here, and that is that what we see with our eyes is not spectra. We do not see spectra, right? We are projecting spectra into a 3-dimensional space given by the activations of the cones, and that then constructs our color space. And the consequence of this is that we can have infinitely many spectra, and here I'm showing you just 3, that have the same appearance to the cones, but are very different spectrally, right? So we call this metameric lights. It's a fundamental fundamental finding, I think, of how we think about the human retina, how we think about light processing, is that we have sort of spectra that will look very different in the spectral domain, but look similar or the same to the human eye. Now, if we want to measure spectra, we have different devices. Here, I've just selected 4 devices that I personally have worked with. They come in different form factors. They come with different optics, different practicalities, and of course, also different price ranges. But ultimately, they all have some technology that converts the light through a diffraction grating, and then you are basically imaging that on a sensor, and then you can read off the spectrum. And it's not only the price that differs, it's also, of course, which conditions they can measure under. So some are more and some are less sensitive. But these are what we call spectroradiometers that measure the spectra of light. There's one important component when we're looking at the exposure geometry, and that is the difference between irradiance and radiance. Okay, when we're talking about, for example, the photopic lux, the illuminance of something, we are talking about something that ultimately is related to an irradiance-like geometry. And this is usually measured in such a way that you have light impinging on a surface, and all that light is captured in some way, for example, using cosine correction. And then the reading it reflects all of these, all the rays of light, as it were, that hit that specific detector. Irradiance-like measurements can be used to turn into illuminance, and similarly, we can calculate the melanopic equivalent daylight illuminance, melanopic EDI. And this is sort of the standard for how we typically consider measuring light when it comes to human responses to non-visual effects of light. When you're looking at other disciplines, so for example, when you look at color science or people working with displays, what you find is that radiance and derived measurements are a lot more common. So radiance measurements are measurements that basically take the light that is coming from one specific solid angle in one specific direction, and from that you can calculate, for example, the luminance. There's a really important difference between these two measures. If you have a homogeneous light source and you move the measurement device closer or further away, it will change the irradiance, but it will not change the radiance if the light source is homogeneous. So if you have a homogeneous screen, for example, that you're measuring from, whether you collect from a 2-degree field or 4-degree field or whatever, doesn't really make a difference. But for irradiance, of course, the closer you are, the brighter it is. And that follows the sort of standard drop-off following the square root, right? So irradiance is always depending, irradiance and derived measurements are always depending on the distance to the source. So where are we? We have now a knowledge on how to measure the light, and we use a spectrometer. Now we want to characterize the light, and for that we use spectrally dependent metrics. I've already foreshadowed the CIE SO26 system, and this is a system for metrology that was published in 2018, which really is the standard for quantifying light. There is an older standard, or an older proposal for a standard that proposes a quantity called melanopic lux. This is not consistent with the standard and should not be used. Similarly, there are no other quantities that are sort of have the level of the standard. So really the CIE system is without a doubt the standard system to use. The nice thing about it is also it's theory neutral, so it doesn't propose any specific interactions between photoreceptors. It just quantifies how much does each of the photoreceptors see. And we call this the alpha-opic system because we have 5 photoreceptors, and alpha is sort of the placeholder for the different photoreceptor types. And so L and M curves combine to make the photopic luminosity curve, which underlie illuminance and lux, and the melanopsin underlying IPRGC responses produce melanopic irradiance and derivatives such as ABI, for example. How would you do these calculations? So this is a little bit of calculation, a little bit of linear algebra ultimately. This is what the data might look like. You have wavelength, you have irradiance from your measurements, you have the spectral sensitivity function, and what you just do is you take a weighted product of the irradiance and the spectral sensitivity function, and then you sum that up across wavelengths. It's a classic sum. In continuous terms, it's an integral, but ultimately it's just a linear algebra calculation where you take one vector multiplied with another in different dimensions. Now, if you don't want to do this by hand, which I hope you don't want to do, then there's calculation tools. We developed LUOX a couple of years ago, which is validated by the CIE. Johannes Zauner, who's now in my team, has produced a tool called Spectran, which is available through the LITG, the Lichttechnische Gesellschaft. And both of these are basically suited to make the calculations to go from spectra into the physiologically relevant metrics and quantities. Now, Finally, I want to highlight a little bit reporting. So stimulus conditions should be reported using a standard and harmonized way. And there are, again, various documents that have proposed things. One is a document from Martine Knoop from 2019, Methods to Describe and Measure Lighting Conditions in Experiments on Non-Image Forming Aspects. There is a paper that myself, Oliver Stefani, Peter Blattner, Claude Granfier, Steve Lockley, and Rob Lucas wrote, which is How to Report Light Exposure in Human Chronobiology and Sleep Research Experiments. And then finally, there's also a CRE technical note, what to document and report in studies of IP-IGC influence responses to light. And we developed through an international consensus process another document, and that's the nLight checklist, the nLight reporting checklist, which really has the goal to kind of standardize and harmonize reporting on stimulus conditions in laboratory studies on the effects of light. And so there's various relatively low-level information that one will need to include, such as the author names, title of the manuscripts. We have protocol-level characteristics that basically describe things like the intensity of exposure, where the measurements were taken, what we know about participant levels, aspects of the light sources, whether it's overhead illumination, whether it's from an emissive surface or from light glasses, things about the type, make, and manufacture of the light source, and if there's any filtering apparatus in a study. So you might imagine, for example, someone, or in a protocol, you might have blue blocking glasses. So this would also be reported here. And then all of the quantitative information. And so what we have here is basically what we think about is the minimal set of light level characteristics. One is the illuminance or the luminance, the spectral irradiance or the radiance, the alpha optic irradiance or the radiance, and then the alpha optic equivalent daylight illuminance or luminance. And so depending on what sort of light source and geometry you have, for example, luminance only really makes sense when you have emissive surfaces. But really with this information, we have a lot of information. And the logic of the checklist is that basically you want to put these measurements somewhere in a supplementary table and basically make them available for posterity so that when someone wants to go back and try to understand, so what did this study do, that all of this information is there and transparently available. This nLight checklist was endorsed by many scientific societies, funders and journals, including SRBR, the Sleep Research Society, and several journals as well. So this is where we are. We have measured light, we've characterized light, and we can report light. And we use different tools for this. We use spectrometers for the measurements. We use reproducible software tools for the reporting, for the quantification. And we use the nLight checklist for harmonized reporting. So now this is all laboratory light exposure, and I already mentioned we want to understand real-world light exposure. In the laboratory, I might have the opportunity to control what participants are doing, what exposure they might get. I might ask them to hold still or put their head on a chin rest, or I control the lighting or have some other way of control. And now, of course, what we're interested in is understanding, well, in the real world, we're never really that controlled. We have certain areas, of course, where we don't have control. One, for example, is office lighting, where typically, you are spending your time in an office that you have not spec'd yourself. And so this means that the light exposure that people get throughout the day is quite variable. And this then poses a specific question, which is, well, if we wanted to understand the real world effects of light, how should we understand this? This is an example of a study of a participant who's participated in a study from done by my master's student, Carolina Guidolin, who was wearing a wearable light logger at eye level. And they were wearing this device for 24 hours for multiple days. And so what you can, of course, they took it off during sleep. But what you can see quite nicely here is that you have quite a spiky landscape, as it were, of light exposures, depending on if you're spending time outside, if you're in the office or not. Things are quite variable. In the background here in blue, we have plotted the daylight potential. So at the same time as these studies, we put a sensor on the roof of our institute to just kind of understand what might light exposure look like if you were on the roof of the institute. And what you can see and what you can appreciate here is, of course, that the photoperiod, so the time that daylight is available, starts a lot earlier than this person's day, who wakes up maybe at 7:30. And of course, this person also goes to bed a little later. And so this is the typical light exposure you might get in ambulatory studies or in field studies. And what we, of course, want to understand, how do we turn these into insights as well? And we broadly know that there's different types of effects. As I mentioned initially, we have disruptive effects of light in the evening and at night. We might have positive effects of light during the day. But most importantly, there's also growing evidence that the light exposure during the day can offset, for example, specific effects in the evening, or change them or modulate them in some way. And so what we really need then is an appreciation and understanding and a measurement of light exposure through wearable technology, so that we really have personal exposure estimation. And so, again, moving from lab to field requires good measurements of light. We coined this term spectral diet a few years ago, which basically is the idea that you have different types of light exposures that vary throughout the day, and we can measure these with wearable light loggers. This is a prototype that we developed here at the MPI. There's now also commercial devices, of course, that can measure this in different form factors with different conveniences. But this is what we're spending a lot of time on, is understanding light exposure in the real world. And so one important thing is, when you now have these patterns of light exposure, how do you turn this into, again, insights? Right here, if in the worst case, if you're looking at this, you have, say, samples every minute. What are you going to do with it? Of course, you can ask, how many times was it within the recommendations of, say, Brown et al. That I introduced earlier? But nonetheless, there's still no sort of agreed upon standard way to condense these time series of light exposure across a wide variety of outcomes into sort of one coherent set. And so my team, led by Johannes Zauner, has developed an open source package called LightLogR, which basically solves this problem for you. It has the ability to ingest almost every data format that exists in the world of light exposure measurements. If you have a light logger, or if you have data that cannot be ingested by it at the moment, please get in touch, because we're happy to spend some time to make sure that our tools can read these data. And so within LightLogR, we have several metrics. And again, there's not really one way that we can think about what the right metric is. And so our philosophy behind this is really to say, well, given that we don't know which of these features of a 24-hour pattern of light exposure drives the non-visual effect, we better calculate as many as we can and then see whether we can do something like a sensitivity analysis. And so here, these are different metric families. Dose, for example, we have duration above threshold, we have threshold for durations, time above threshold, a common one, but also things that are kind of more from the circadian field, such as intradaily variance and intradaily stability. And so we have this really armada of different different quantities we can calculate here, and do this all through this very convenient interface provided by LightLogR. I want to give you an example of why we might want to use wearable light measurements. And so what we've done recently was examine light therapy lamps. And so this is a protocol where we had two people sitting in the same room. One received light therapy, the other one didn't. And so it was just overhead lighting for both of them. And then one person sat in front of a light therapy lamp. Both wore light loggers around their neck as a lanyard. And so here in blue, you see the trace of the person who had the light therapy lamp. And here in yellow, you have the trace of the person who had no light therapy lamp. And what you can see quite nicely here is, of course, how these light exposures differ. I do want to point out that for neither participant, the light exposures that they would get are stable, right? It's quite variable. You have a little bit of variability. And of course, this depends very much on how your body is positioned relative to the light source. I showed you already earlier this light logger here, which is quite big, worn on the head. Ultimately, whether or not you should use a light logger that's worn on the head or worn on the chest or worn on the wrist depends on the research question. We now have just completed a data measurement campaign where we had looked at 3 locations, and we're just finishing the analysis of these. But what you can do now, right? So this is the time course. What you can do now is calculate the dose over time. So this is the cumulative dose that someone will have received throughout the day. We calculate this as the melanopic EDI dose in lux hours, or melanopic EDI lux hours. And of course, what you can see quite nicely here is the steepness of this curve depends on whether you get the therapy lamp or not. And again, these are analyses done with light log R. So what we can calculate are basically statistics of light exposure. And of course, if you're in the therapy lamp condition, then the exposure will be a lot higher. And you can see quite nicely the differences here. And so there's value even when you're looking at things like light therapy to basically have methods to track and measure light exposure as it happens. As I mentioned, we have this tool available, LightLog R, that I think takes the measurements or some of the measurement principles and the quantification principles that I've described to really an open source software package that is supposed to be as convenient as possible. We also offer training on this. We had a beginner's course that is now available on YouTube, and we have an advanced course. So if you're interested in understanding how you can use analytics and open source tools for the analysis of these light exposure data, then make sure you follow the QR code because there's a registration link. And so really what this brings us to is an understanding of light exposure in the laboratory that we should quantify in ways that are reproducible, but also as we are moving around and moving from field studies to lab studies, from lab studies to field studies, that there are, again, reproducible ways that we can capture and track light exposure. And with that, I'm happy to close. This is sort of, for me, has been an interesting journey to go from being very much a visual neuroscientist to being a circadian scientist, and nonetheless trying to integrate our knowledge of what's happening at the front end of the circadian system to then the responses. And I believe that having a physiologically relevant framework, such as from the one from CIE SO26, is exactly where we should be to make sure that the light measurements we do and the exposures that we infer and the health outcomes that we then relate these to are computed in a space, right, that is relevant to actually how light is processed in the eye. So with that, thank you again for inviting me, and thank you as well for your attention.